Thank you for the introduction. So, yep, my name is Brian, and I'm presenting my summer research that I did this past summer at the Stanford University School of Medicine. Um, the project title is named An Investigation of B cells and age-associated B cells during chronic CMV infection among transplantation patients. So that's quite a convoluted title, and I'll be going through the background to make sure everybody's really caught up to speed. Because being in immunology, and especially in transplant immunology and viral infections, is a very convoluted system to actually study. So there must be very strict controls are actually established in order to actually take away values. So, you know, immunology as a very broad sense is just the study of our immune system, right? And then there's two very main branches of immunology, right? Innate and adaptive, right? So in our lab, we really focusing on the adaptive, which is what I'll be basing our background off of. And in the adaptive immunity, right, this is where you have antigen specific players that are coming to evolve, right? And one of the key players that we're very interested in are the B cells, right? And B cells are these lymphoid cells. They differentiate into plasma cells, which then create antibodies. And antibodies are key to actually establish our humoral immunity, right? And a bunch of key, very being very brief, bunch of very key functions of the B cells is that they're antigen presenting and they're actually able to recognize antigen with their BCRs. So to go into our specific cell of interest is what they're called age-associated B cells. They're a subset of these general B cells, and they're actually quite recent in literature, right? We don't really know too much about them, and the heterogeneity of the definitions of B cells is actually quite diverse, just because people are still yet to come to consensus of what is an age-associated B cell. Um, so researchers have noticed that as one ages, there's a particular, distinctly phenotypical cell that expands in number, and we don't exactly know why, right? And that specific B cell with this distinct phenotype, which is it's expressing the transcription factor of PBAT and D11C, and they lack CD21 for expression. Um, so the 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 age-associated B cells are known to be highly inflammatory. They have decreased antibody production, right? And the associated two factors lead to decreased effector function, right? So, with an increase of age-associated B cells, researchers are not quite sure whether or not that correlates to immunosenescence and immunodysfunction, or if age-associated B cells actually have immune function when it comes to age. Whether or not age immune in our aged immune system, whether or not age-associated B cells are actually contributing or helping. Because there might be a hidden benefit to what age-associated B cells are actually contributing to our immunity as we age, right? At the time, not exactly sure what it is or why it is doing, right? But we do know that they do produce inflammatory cytokines and autoantibodies, which are known to be harmful. Um, and the tricky thing with age-associated B cells is, along with being accumulating with age, they also have been known to be enhanced in cases of autoimmune disorders and chronic viral infections, which plays very well into the systems that we're studying with CMV infection and transplantation, right? And to go very briefly into T cells, these are for your cell-mediated immunity, and then they're very two general types of T cells: CD4 T cells, the helper T cells, and cytotoxic T cells, CD8s. So to explain why we're looking at CMV, CMV is a very common viral infection with super high prevalency rates, and I would bet that at least one person in actually this call right here probably has CMV. But I wouldn't worry too much about it, just because in healthy patients, CMV doesn't actually do anything. It's what they call an opportunistic infection, where it only happens it only causes problems when you are facing cases of immunodeficiency, right? So in cases of ADHD or HIV, um, HIV, or in our case, transplantation, where you have to take immunosuppressive drugs in order to prevent allograft rejection. Um, and the immune system has several ways of dealing with CMV. Likewise, has one of the largest DNA genomes that we've ever discovered in a virus, has multitude of ways of actually evading our own immune system, right? Such as MHC1 decreased expression, right? MHC2 decreased expression um, can decrease NK activating ligands, and then they have very high antigen variation. So all those factors really make CMV a very hardy infection, and chronic CMV infection wreaks havoc on our immune systems, especially in transplant patients. So, with the immunosuppressive medication combined with chronic CMV infection, that creates many immunological downstream effects that, as a science community, we have really yet to really study or understand. And from our lab, specifically Dr. Musman's lab, we have predominantly investigated T cell behavior in the past, right? And we found T cell behavior such as we see cytotoxic T cells develop a seemingly cytotoxic phenotype, which really shouldn't be the case. We see huge expansions of cytotoxic T cells. We see premature aging of the immune system and premature aging of T cells in general. And then we see an evolution of the clonality of T cells as well, right? And all of those are not really supposed to happen, and yet we don't really understand why. So the next iteration of this project and of our lab is that we know that B cells and T cells interact extremely closely together, and they're actually crucial for the activation of each other. So therefore, a very logical conclusion that we had was that T cell dysfunction leads to, or is very closely related to, B cell dysfunction, which is why we wanted to study age-associated B cells and B cells in general in this system, right? So the study aims to investigate the potential transformation and shift of B cells in the context of CMV infection among transplant patients. Right. So here's a bunch of these research questions, right? What are these functional properties of these age-associated B cells in CMV positive transplant patients, and how do they contribute to immune dysregulation, right? Can the measurement of age-associated B cells and B cells serve as potential biomarkers? And does the presence of CMV-specific antibodies or memory cells correlate with expansion of age-associated B cells? And all these questions really sought to answer throughout, not not just with my summer research project, as this project continues to evolve over time as we learn even more. So our hypothesis is that we hypothesize that chronic CMV infection in transplant patients leads to an expansion of age-associated B cells and B cell populations. And that's a very logical conclusion, because based off literature, we know that age-associated B cells increase with age, but they also increase with viral infection and autoimmunity. Right? Both of those cases, autoimmunity, you could make a case that your transplant, your allograft, is an autoimmune disorder. Right? Not really, because technically that is a foreign tissue, and that constant viral stimulation, antigen stimulation of the CMV and allograft would um, logically cause an expansion of age-associated B cells. So the materials and methods. Flow cytometry was utilized as the main method for cell population analysis. Um, and so just to go very briefly in flow cytometry, it's a technique used to analyze cells based off specific light scattering and fluorescence. Um, and looking at each individual cells grants us a very huge amounts of data in order to actually analyze. Right. And this is my experimental timeline of where I started and where I really ended. So huge parts of the summer was actually doing the panel design, right? And then once we established the panel, which is what we were actually able to do at the end, we wanted to move on to testing more and more patient samples. And here's our very general cell panel. very general. You do the CD3, CD14, 16 negative gates to actually gate out all the non-B cells. You do CD19 positive gate to gate our positive B cell marker. CD21, CD11C is TBAT, our age-associated B cell like definition, so to say. So you want to look at this is our gating strategy. I'm not going to go too much into it, but just you do the general lymphocyte gate, followed by double single gates, and then a live gate, and then you follow the flow cytometry binary to actually remove subsets of cells that are not your cells that you're most interested in. And then you end up with your age-associated B cells, in which those are the ones you want to analyze the most. And our anticipated results is that just because a huge majority of my summer was actually established a flow cytometry panel, right? Do the antibody titrations, the compensations. We were actually able to generate viable data, right? But I have some very test cases at which we would utilize this type of process in order to actually test what our data would look like in the future as the lab actually progresses. Like I said earlier, we do anticipate seeing the observation of an expansionary effect of age-associated B cells and B cells, just because your body needs to, in order to establish your immunity, your body needs to constantly increase antibody production, right? And logically, it really makes a lot of sense why age-associated B cells and B cells in general would increase as CMV infection and transplantation rage on. So, the utilizing and actually collecting all this question data actually gives a huge amount of data, and shifting through it one by one with individual gates is actually quite tedious and not a very good use of my time, in my opinion. So we really want to utilize dimensionality, so-called dimensionality reduction algorithms, so known as TISNE algorithms. And essentially what these algorithms do is that they cluster the cells based off fluorescent activity, based off similar fluorescent activity on this arbitrarily defined plot here, shown on the right. And the idea is that two plots that are close to each other on the plot exhibit similar fluorescent activity, right? And by doing so, populations are actually be able to group together. That allows you to find hidden patterns in populations that you would not have been able to easily find without such a dimensionality reduction, right? So I decided to run this with my data, and I utilized two different patient samples, one being CMV positive, CMV negative. I ran the normal age-associated B cell panel along with additional markers, including the immunoglobulins, IgG, IgM, and IgD, CRX3, which is a chemokine receptor, CD27, which is a cell memory marker, and an immune checkpoint receptor known as TIGIT digit. And the associated plot looks like this, right? This itself does not give you too much information until you overlay the two. The red being CMV negative and then the blue being CMV positive, right? And this gives you a good idea of what the populations look like compared to each other and compared to actually other age-associated B cells within each sample, right? So what we were really curious about was we see certain populations of age-associated B cells that only occur in one sample or the other. In this case, only in the CMV positive section, which calls to question, why is that the case? Because that really shouldn't be the case, right? The age-associated B cells should all be the same compared to each patient. And then if you look at this population right here, this one only shows up in the CMV negative population. So that calls us to investigate further. So we run another algorithm. It's called a cluster analysis algorithm to see actually what those clusters look like. And then it just becomes the basic busy work of looking at, we define the previous defined cluster overlaid onto our double sample map. And then we notice that, so we see huge overlap between all three samples, which is what should be the case. Then we have certain cases where, like this, which is also quite homogeneous. This is triple it appears in all three cases, right? But we have situations like this where the sample only, the cluster only appears in one sample or the other. And if you want to shift through the data, essentially that's what we're looking for. Is we want to see populations of age-associated B cells that are only in that consistently appear in one type of population over the other, and that allows us to investigate further what the actual overall function of those age-associated B cells are doing, and what the age-associated B cell population looks like compared to CMV positive, CMV negative. So the very broader implications is we want to see the interplay between these B cells, CMV, and immune evasion, right? Knowing that because it's quite complex, and considering the virus's ability to actually dodge the immune system, um, right? By understanding this interplay, we're able to understand how CMV is able to manipulate the immune system, and then how age-associated B cells might be involved in that process. We want to have more insights in chronic viral infection. Um, we want to see the effects on B cell populations, and then ultimately, right, because this is a clinical study, we want to see implications for transplant patients, right? We want to understand the impact of CMV on our immune system, and we want to utilize that information to improve transplant outcomes and manage post-transplant complications. So yeah, I just want to say a very quick thank you to the Paper Trail Symposium, my research lab at Stanford. Very grateful for this opportunity to present my research. Thank you.
Thank you, Brian, so much for your presentation. We were very happy to have you today. We're out of time really for questions from the audience. However, please feel free to DM Brian through the Zoom chat, or contact information will be available to email students if you have additional questions about the research. However, we do want to hear about what your future plans are. What are your interests in potentially doing research in grad school or beyond?
Yeah, I'm very interested in doing very immunologically focused research in grad school. So
Awesome.
looking at viral infections and all that.
Perfect. Well, thank you again for being here today with us.